Welcome back, Dr. Linder here. Okay, what I want you to do before I move on in this lecture to the steps that initiate a contraction and steps that end a contraction, what you should do is review some of the basic terms that are involved. You should be very familiar with the contractile proteins. Those are myosin and actin. You should be familiar with the regulatory proteins, troponin and tropomyosin. You heard me in a previous lecture refer to tropomyosin as the rope and troponin as the staple. Well, what was the staple remover? That was calcium. Where was the calcium stored? In the sarcoplasmic reticulum. So you should know the regulatory proteins, the contractile proteins, and the structural proteins. Okay, so now we can say the orange ropey structure was the tropomyosin and the yellow staple is the troponin. We want to make sure that calcium or the staple remover removes troponin so that the tropomyosin shifts, exposing the binding sites so that the myosin heads can attach and create a contraction. So let's look at the steps that take place. On this side are the steps involved with a contraction, and here the steps that end a contraction. And that goes along with this slide, steps that initiate a contraction and steps that end a contraction. So there's about 5 steps in the left side and another 5 steps on the right side. So if we look here, we see this is the neuromuscular junction. What do we see here at the very end of the axon is an axon terminal. And at the very end are the synaptic end bulbs, which have these vesicles that contain a neurotransmitter. What is the name of that neurotransmitter? The name of that neurotransmitter is acetylcholine. What tells acetylcholine to release through exocytosis? Well, that would be calcium, the influx of calcium. Calcium tells the neurotransmitter to exit through exocytosis. Now it's in the synaptic cleft. Now it meets the end plate of the muscle where it binds. So after it binds, the sodium channels open. Sodium rushes in. It goes down the transverse tubule, that invagination, and creates an action potential. The action potential reaches the transverse tubule, which butts up against the sarcoplasmic reticulum. The sarcoplasmic reticulum stores calcium. It releases it to the sarcomere. The sarcomere has these 2 contractile proteins, actin and myosin. Somehow this calcium is going to remove the troponin, remove the staple, so the tropomyosin shifts out of the way, and when it does, just like an elbow, this looks like an arm, an elbow, and a wrist, and a hand. Think of it that way. The elbow joint, the wrist joint, and the hand. The elbow bends, and now the hand can attach to the binding site. And when it binds to the binding site, now the hand can flick it and create a contraction. But that's just one flick, right? Imagine having your hands on a tug of war rope. When you pull the rope in, don't you have to release the rope and move one hand in front of the other to pull again, and then release a hand to pull on the rope to pull again, and then release a hand, put it in the front, and pull again, okay? So that has to happen. Each time you release the myosin head, it requires ATP, or energy, each time you do that. Okay, now what's happening over here? How does the muscle contraction end? Well, what was it that initiated it? Well, we had this acetylcholine that was in the synaptic cleft. Well, let's get rid of it. So how do we get rid of acetylcholine? We get rid of acetylcholine by using an enzyme called acetylcholine esterase. Acetylcholine esterase is an enzyme that degrades and breaks down acetylcholine. We don't need it anymore. Well, if there's no more acetylcholine, then that means there's no sodium rushing in, there's no excitation, there's no more calcium that's going to be released, so calcium gets put back in its storage container in the sarcoplasmic reticulum. If there's no more calcium being released, then there's no more contact between the myosin heads and actin. So the binding sites are covered again. And the contraction ends, and it goes back to relaxation. So you can review those steps. They're here, you can pause it, but this is basically everything that takes place, okay? So the events that take place, some of the highlights: the voltage-gated calcium channels in a neuron synaptic end bulb opens, resulting in an influx of calcium, right? The calcium moves in the synaptic end bulb and says, hey, neurotransmitter called acetylcholine, you're going to move to the end through excitosis, and I want you to release that acetylcholine into the synaptic cleft. Now the acetylcholine, NT, is the neurotransmitter, acetylcholine, binds to the sodium channels. The sodium channels open, causes an influx of the sodium into the muscle. That causes the depolarization or excitation of muscle, and it then results in the calcium release from the sarcoplasmic reticulum. When that sarcoplasmic reticulum releases the calcium, you get a muscle contraction. When you don't need the muscle contraction, then the neurotransmitter acetylcholine is broken down by acetylcholine esterase. Okay? So the myosin is going to pull on the actin, causing that thin filament to slide inward. And thanks to the structural proteins, nebulin, right? Remember nebulin, dystrophin, there's a transmission of the force throughout the entire muscle, resulting in the whole muscle contraction. How neat is that? So if we were to look up here on the top left, we see that the binding sites are exposed. Well, how did they become exposed? Well, you see calcium pulled the troponin out so tropomyosin can shift, and now these binding sites on actin are open, so this myosin head extended up, making contact to the binding site on actin, right? This is like the elbow joint extending up, the wrist joint is straight, and the fingers are gripping on. Think of it that way. But then the wrist flicks. Look at the shape of the wrist joint here compared to the wrist joint here. It flicks it, creating what's called a power stroke or a contraction. But after this bends and moves, it has to detach only to grab on again. Now, notice what we see here at the myosin head. We see ADP. What does ADP stand for? Adenosine diphosphate. Adenosine diphosphate. So it's an adenosine di with two phosphates. And hanging out in the wind here is a third phosphate. Usually this phosphate hangs on to a protein called creatine. So usually it'll be CP or creatine phosphate. So creatine phosphate, or this phosphate, has the ability of joining forces with ADP. Now, what do you think is going to happen if you have two phosphates and a third phosphate attaches to it? Well, it's called ATP. Now you have adenosine triphosphate. That's the union of this third phosphate to ADP. When it interacts, we now have energy to detach the myosin head. Detachment takes energy in the form of ATP. Once that's done, we go back to ADP and a phosphate again. Okay? The body doesn't like to keep ATP hanging around in the form of ATP if it's not being utilized. So it'll store some of those phosphates as CP, creatine phosphate, which can be utilized for immediate energy, fast bursts of activity. Okay, now we know that detachment requires energy because if you've ever watched an episode of like CSI Miami or any when forensics show up to a murder scene, sometimes you see like the person in the state of rigor mortis, where the body is in a contracted, shortened state. Why is that? Well, if the body's in a shortened, contracted state, the myosin heads weren't able to release. Why couldn't the myosin heads release? Well, the person's dead. If they're dead, the Krebs cycle isn't working. They can't make ATP. Okay? Okay. We'll take a break here. When we come back, we'll talk a little bit about muscle metabolism, which does involve a little bit of creatine phosphate that we mentioned before in this video.